acceptor, phytin, and then to plastoquinone. You really need to know plastoquinone. From plastoquinone, they go down a cytochrome chain or an electron transport chain and back to photosystem 1. The electrons that leave photosystem 1 are passed to ferredoxin. Again, it's an iron sulfur protein. And ferredoxin can pass the electrons to NADP, reducing it to NADPH plus H+. The only difference between NAD and NADP is that NADP has a phosphate on it. Still, this energy capture is a reduction reaction, not a phosphorylation reaction. So energy is captured both as ATP and as NADPH plus H+. Again, because the reduced coenzymes have a higher energy potential. There's our energy capture, ATP and NADPH. Now, non-cyclic photophosphorylation, if the organisms are doing non-cyclic photophosphorylation, they get to capture more energy because they get to capture energy both as ATP and NADPH. If the organisms are doing cyclic photophosphorylation, they only get one. It turns out that algae and cyanobacteria can run a non-cyclic pathway, which I'll show you in a moment. The other thing that's talking about here is where is this all taking place? So photophosphorylation, the molecules that are shown in those diagrams are associated with membranes. And the membranes are thylakoids. Thylakoids are the inner folded membranes in chloroplasts, but thylakoids are also the folded membranes of cyanobacteria. So whether we're talking prokaryotic cells or eukaryotic cells, the membranes are thylakoids. Thylakoids means sac, and these are little sac-like, well, they're membranous sacs. Anyway, here you see the membrane diagram where protons are being moved by the cytochromes into the thylakoid sac. So they're moved into the interior. So the cytochromes are moving moving protons in here. Their electrons are passed from one cytochrome to the next. Protons are moved across. That generates the proton motive force, and then they come back through ATP synthase. Again, every diagram that you see like this, these are all oversimplifications because there are way more molecules than what you see here. So again, what you often don't see in these diagrams, if you're looking at the diagrams that are in the lecture syllabus where you just see the antenna array and then you see arrows, one arrow going to plastoquinone and then another arrow going to ferredoxin, you see electrons going from plastoquinone back to photosystem 1, traveling through a cytochrome chain. What you don't see is that the cytochromes are in the membranes and that those are going to be moving hydrogen protons through the membranes. So you just have to remind yourself that. So there you see the chlorophyll molecule passing electrons and then also moving protons through the membrane. So you have to have a proton motive force. Again, this is very much like oxidative phosphorylation. You have to get a concentration of hydrogen protons on one side of the membrane, then they come back through ATP synthase, and ADP plus phosphate is converted into ATP. As far as I know, it's always 3 protons through ATP synthase to make 1 ATP. It doesn't matter whether the organisms are using photophosphorylation or oxidative phosphorylation. That it has to be 3 protons going through to make 1 ATP. And again, ATP is our energy currency. Now, the organisms have a slight problem because the electrons that have left photosystem 2 don't come back. Electrons go from photosystem 2 to plastoquinone to photosystem 1 and then off to ferredoxin and to the coenzyme. Photosystem 1 is constantly getting new electrons that are coming from photosystem 2, but photosystem 2 doesn't get any electrons back if we're running a non-cyclic pathway, so they need to pull electrons away from something. And what you see down at the bottom of this diagram is where the electrons are coming from. They're going to come off of water. So if you recall, algae and cyanobacteria are oxygenic. And they are oxygenic because they can split water molecules. If you pull 2 electrons and 2 hydrogen protons away from water, then you're going to get oxygen, molecular oxygen. So when we say that the pigments in photosystem 2 have to replace their electrons, they're going to pull them away from water. The result is going to be molecular oxygen. You don't need to worry about balancing equations, but if you look at it, it's like, yeah, okay, if you had 2 waters minus 4 electrons and 4 hydrogen protons, you get O2, molecular oxygen. So when you see organisms growing in a water mat like this, these are samples of algae. The bubbles that you see with them, that's oxygen, and they're producing it. Now, if algae and cyanobacteria run a cyclic photophosphorylation, which they can do, then the electrons that pass from photosystem 1 go to ferredoxin, go through a cytochrome chain, and then come back to photosystem 1. This is an alternate form of photophosphorylation that these organisms can run. What this tells us is that algae and cyanobacteria are not always going to be oxygenic. They're only going to be oxygenic if they're running a non-cyclic pathway. If they're running a cyclic pathway and the electrons come back to the pigment, then they're not going to be pulling electrons from water. So this is showing you a cyclic pathway. They can still make ATP, running electrons down the electron transport chain, the cytochrome chain, but they cannot reduce the coenzyme NADP. So they only get to capture energy as ATP. Notice also that when photophosphorylation is cyclic, you can't split water, they're not going to form oxygen. So again, just because organisms are potentially oxygenic doesn't mean they're always oxygenic. When's the other time when algae are not oxygenic? At night, exactly. We talked about this relative to eutrophication and why eutrophication can be so damaging, because if you have lots of algae in the water, they are respiratory organisms. They use oxygen. At night, they can't make it because there's no light, and that's why they suffocate the fish. So the other time they can't make oxygen is at night. So now we can go back to how does photophosphorylation relate to photosynthesis? Because this whole section, the beginning part of it, it said it was about photosynthesis and biosynthesis. We've only talked about photophosphorylation so far. So notice those two words, photophosphorylation in green, photosynthesis in tan, I guess. Please do not use those interchangeably because they don't mean the same thing. Photophosphorylation has a very specific meaning. It's the production of ATP using light as an energy source. Photosynthesis, on the other hand, suggests that the organisms are actually going to synthesize organic compounds. Photosynthesis has to do with making carbohydrates. Photophosphorylation doesn't. So photosynthesis, light synthesis, it's building reactions. It's a process that allows organisms to make sugar, supposedly, and oxygen. But we've seen here where sometimes they make oxygen and sometimes they don't. But if they're going to make sugar, they're going to have to run a pathway that we haven't seen yet. This is an equation that you'll sometimes see where organisms can take carbon dioxide and water, and using light energy, they can convert that into sugar, C6H12O6, and oxygen, O2. Well, this is convenient. It's a handy equation, and you'll see it very often in general biology textbooks, but we've just seen that it's not always true, because algae and cyanobacteria can run photophosphorylation and not produce oxygen. The question is, can they make sugar and not produce oxygen? And the answer is, oh yes, they can, because the production of sugar isn't tied to oxygen production at all. Again, think about molecules that are significant to one another. CO2 in this equation is going to end up here. So we've got 6 CO2s. Those 6 carbons are going to be these 6 carbons. We're going to use carbon dioxide to make sugar, and we're going to use water to make oxygen. Using water to make oxygen, that's happening during photophosphorylation. This other process we haven't talked about yet. So this equation is only sometimes accurate. If you're talking about these organisms, it's never accurate because these are anoxygenic phototrophs. So these are the green and purple sulfur bacteria. Actually, all of those are purple sulfur bacteria. Those guys can carry out photophosphorylation. They never produce oxygen as a byproduct. They're anoxygenic. And then we've also seen that although algae and cyanobacteria and green plants produce oxygen, they don't always produce oxygen. If they're running a non-cyclic photophosphorylation process, then they don't, because they don't split water. Excuse me, they're running cyclic. The only time they split water is if they're running non-cyclic. So, the other thing is, we talked about other nutrient categories. So although photoautotrophs use light energy and inorganic carbon as indicated by that equation, photoheterotrophs don't. Photoheterotrophs use light energy, but they're using organic carbon. And chemoautotrophs don't either, because chemoautotrophs are using strictly inorganic compounds. They can still produce sugar, and they're not using light. Anyway, all of those guys are prokaryotic. So what is photosynthesis then? I said it's not the same as photophosphorylation, so what is it? Well, it's a two-part process. The first part is photophosphorylation, using light energy and capturing energy in high-energy compounds, making ATP and NADPH. The second part of it has to do with capturing carbon. We're going to look at one way that organisms can do this. It's called the Calvin-Benson cycle. It used to be just called the Calvin cycle, and then they added Benson, so now it's Calvin-Benson. And there's a third name out there that you'll see sometimes, so sometimes this is called the Calvin-Benson-Basham cycle. I think I stuck it in the notes someplace. Yeah, anyway, these are also sometimes referred to as the light-independent reactions. Some textbooks call them dark reactions. Well, they don't have to happen in the dark. They just don't require light. So they can happen in the dark, it doesn't matter. But basically, this is an anabolic pathway, so it's a building pathway. And it's going to allow organisms to take inorganic carbon, carbon dioxide, and use it to make sugar. So one more metabolic pathway you guys get to look at. The enzymes that are used to run this pathway are associated with the stroma of chloroplast. So if you look at a chloroplast, the inner fluid-like material is the stroma, equivalent to the matrix of a mitochondria. The stroma is the inner part. If we think about chloroplasts as being prokaryotic cells, then what would be the stroma would be just cytoplasm. On the other hand, if prokaryotic cells are fixing carbon, what do they use? Hint, they're inclusions. You remember the inclusions that prokaryotic cells have that contain enzymes involved in fixing carbon? Carboxysomes. Right, carboxysomes. So they don't just have their enzymes floating freely in the cytoplasm. Their enzymes are concentrated. Apparently this has to do with the fact that the primary enzyme that they use is not terribly efficient. And so what plants do is kind of congregate the enzyme inside their chloroplast. And if we're talking about bacteria, they concentrate the enzymes inside of carboxysomes. So there we see a chloroplast with its thylakoids. And here we see the inside of a carboxysome. Carboxysomes have a structural layer around the outside, but it's not unit membrane. That is, it's not lipid bilayer with integral and peripheral proteins. I think it's just a protein layer. As you can see the green inside, that's the concentration of enzymes inside the carboxysome. Now, for this pathway, I'm only going to ask you to learn one enzyme. This pathway is somewhat complex, the Calvin-Benson cycle, and probably the most important enzyme is this ribulose bisphosphate carboxylase oxygenase. Fortunately for you guys, it can be abbreviated to Rubisco. And then if you, I don't know if you guys ever listened to those commercials about Nabisco cookies. It kind of sounds like Nabisco. Anyway, Rubisco. If you use the acronym or the abbreviation, please do it as shown here. It's a capital R, small u, capital B, small I, small s, capital C, capital O. Rubisco. sometimes also called RuBP, but I like Rubisco better because it really tells you what it is. Ribulose bisphosphate carboxylase oxygenase. This is kind of like the pyruvate dehydrogenase complex in that it has multiple things that it does. So it is a complex and it's going to do more than one thing. We're going to concentrate on the one thing, which is fixing carbon. So here's a kind of diagrammatic representation of it. That doesn't tell you much. Hardly anything, in fact. However, the name ribulose bisphosphate carboxylase tells you that what it's going to do is take an inorganic carbon dioxide, and stick it onto a 5-carbon sugar, which is ribulose bisphosphate. So Rubisco is catalyzing the reaction, binding inorganic carbon, CO2, with a 5-carbon sugar called ribulose bisphosphate. At the same time, or very quickly thereafter, the 6-carbon compound that is formed is oxygenated, and then it splits in half. And you get 2 3-carbon compounds very quickly. So the 6-carbon compound that forms is basically unstable. This diagrammatic representation is showing you CO2 coming in here. Again, the carboxylation is what's significant. We're going to put this carbon onto a 5-carbon sugar. We're pulling inorganic carbon in and sticking it into an organic form. It's binding to an organic compound. This, once it's a 6-carbon compound, again, oxygen is bound with it, and then it splits in half. And you get 2 phosphoglyceric acid molecules. So you just have to know Rubisco, and that it's binding carbon dioxide to ribulose bisphosphate. That's the most important step. That's the beginning of the Calvin-Benson cycle, or Calvin-Benson-Basham cycle. So you get this highly unstable 6-carbon compound. It splits in half into 3 carbons, and then, so if we think about this, this pathway is ultimately going to release organic compounds, which then the cell can use to build other kinds of things. Very often, when you see the diagram, what you see is Calvin-Benson cycle spinning off glucose or spinning off fructose. Both of those are 6-carbon compounds. Apparently what really comes off of the Calvin-Benson-Basham cycle are 3-carbon compounds. But then 3-carbon compounds can be put together to make 6-carbon compounds, and those can be put together to form polysaccharides, etc., etc. So this is kind of the beginning of the building process. This is a diagrammatic representation of Calvin-Benson cycle. Again, know this enzyme, Rubisco. Bringing in CO2, sticking it onto ribulose bisphosphate, forming a highly unstable 6-carbon compound that immediately splits into phosphoglyceric acid. The other thing that you need to know about this, okay, so you need to know what it is. Calvin-Benson cycle is an anabolic pathway. It's a building pathway. So organisms are pulling in inorganic compounds, CO2, and they're going to use this pathway to build organic compounds. Ultimately, they're going to get to build sugar this way. Because it's anabolic, it requires energy. Instead of releasing energy that can be used to make ATP, this pathway requires ATP. It requires ATP up here at the top. These sugars are phosphorylated, ATP is used, then the sugars are reduced, and that's going to require another high energy compound, NADPH plus H+. There's some rearrangement. We pull out the 3-carbon compounds. The rest of the cycle goes around. Another ATP is used up here. Basically, think of this as ATP and NADPH are driving this pathway. Those are the energy sources. I'm not going to ask you to diagram this pathway. Just think of it as a cycle. You're pulling CO2 in at the top, and you're pulling out organic compounds down at the bottom. If you follow this around, you pull in 6 CO2, you make 6 carbon compounds, which is 12 3-carbon compounds. They get phosphorylated, they get reduced, they get pulled out down here. So we're going to pull out 2 3-carbon compounds, and then we go back around with 10 of these and phosphorylate those again, and then we get to find another carbon, and this just keeps going. So it's anabolic, it's driven by ATP and NADPH plus H+. I'm never going to ask you to draw it, but I might ask you to define it. So again, it's anabolic, it's going to require energy. It's used to build organic compounds. Basically, as I said, you'll see this described as generating 6 carbon compounds. Well, okay, you're going to be able to do that. If you make 2 phosphoglyceraldehydes, you can put those together to form glucose, which can also be used to form fructose or any other isomer of glucose. So it's an anabolic pathway, energy provided by ATP and NADPH plus H+. The other thing you need to know about is where does it occur? Again, in the stroma of chloroplasts or in carboxysomes. So, the Calvin-Benson cycle is tied to photophosphorylation because photophosphorylation provides the high energy compounds, the ATP and the NADPH plus H+. So they are connected, but at the same time they're separate. And if organisms have some other mechanism for making ATP and reducing NADP, they could run a Calvin-Benson cycle without having to do photophosphorylation. And that's what chemoautotrophs do. We rarely talk about those guys. This is just showing the thylakoid, showing that when hydrogen protons are moving across membranes, they're going to the inside of the thylakoid, and then when they go out, ATP is going to be made. So this is showing photophosphorylation happening in this section, and then the Calvin-Benson cycle happening in this section. So the high energy compounds produced here, and someplace else, up here, these high energy compounds are used to drive this pathway. So, and as I said, the products are typically identified as sugars. They're initially 3 carbon compounds. They get put together to form glucose or fructose, and that's fine. Now, what we're going to do next is talk about what happens, what other kinds of things can cells make, because this section is talking about biosynthesis. So, photosynthesis, that is the light-independent reactions of photosynthesis. Calvin-Benson cycle, that's just one way that organisms can fix carbon. There are other pathways organisms can use, and you guys don't have to know them. So you're just being introduced to a particular type of metabolic pathway. I believe it's the most common. And I read someplace that ribulose bisphosphate carboxylase oxygenase may be one of the most abundant enzymes on the planet, because there are so many organisms that are photoautotrophs, and they're all using it. But again, that's not the only pathway organisms can use to fix carbon. It's just the only one you have to know. Well, okay, so if they can fix carbon, what other kinds of things can they build? Well, if they can make 3 carbon compounds, again, you can put 2 3 carbon compounds together to form a 6 carbon compound, and then you can start sticking 6 carbon compounds together to build starch, polysaccharides. But because glucose can be catabolized via glycolysis and the Krebs cycle, there are multiple intermediates along the way that can be used to build other kinds of molecules. So we're going to look at this just very briefly, kind of biosynthesis in general. So think about glycolysis in the Krebs cycle then. Glycolysis is from glucose to pyruvic acid. We were looking at it initially from the standpoint of energy capture, organisms making 2 ATP. But along the way, there were all these organic intermediates, and those things can come out and be used for other things. The same is true of the Krebs cycle. We talked about the Krebs cycle as being catabolic. It's breaking down organic compounds and releasing energy. But the intermediates of the Krebs cycle can be used to build other things. And many of the reactions of the Krebs cycle can run in reverse. And so if you really think about what the Krebs cycle is, it's an amphibolic pathway. It can run either as a catabolic pathway or as an anabolic pathway, where things can be taken out of it and used to build. So if you run into that term sometime, don't be surprised. Amphibolic means both ways. So the triose sugars produced via the Calvin-Benson cycle, they can be used to make fructose or glucose. Talked about that. These hexose monosaccharides can be used to make disaccharides, trisaccharides, polysaccharides, what have you. If you look at the step in the glycolysis pathway where you have the molecule dihydroxyacetone phosphate, that was after the fructose 1,6-bisphosphate split, and we ended up with PGAL, phosphoglyceraldehyde, and dihydroxyacetone phosphate. Well, that DHAP, dihydroxyacetone phosphate, take the phosphate off and it can be used to make glycerol. If you make glycerol, then what kind of organic compound are we heading to make? A triglyceride. We can make a triglyceride. All we have to do is connect some fatty acids to it. So this is showing where that's made. Fructose 1,6-bisphosphate splits. You get glyceraldehyde triphosphate and DHAP. The other thing that we looked at was how we made acetyl coenzyme A. So acetyl coenzyme A was made between glycolysis and the Krebs cycle. The enzyme pyruvate dehydrogenase pulled the hydrogen off, well actually it pulls the carboxyl group off, takes the hydrogen from that, sticks it on NAD, and then the 2-carbon remaining unit is bound with coenzyme A to make an acetyl-CoA. Well, those 2 carbons could be used to build a whole chain of carbons. You can build fatty acids that way. So just, again, because acetyl coenzyme A is a high energy compound, rather than sticking the 2 carbons onto oxaloacetic acid and making citric acid, you can stick the 2 carbons together and just keep building long chains of 2 carbons until you get fatty acids. And if you put the fatty acids together with, so there's our acetyl coenzyme A, sorry. If you put the fatty acids together with the glycerol, you get that, and that's a triglyceride. If you put 1 glycerol molecule and 2 fatty acids together, what do you get? If you stick on a phosphate group, a phospholipid. So we can build those things too. And if you look at some of the intermediates, some of them come from the glycolysis pathway and others come from the Krebs cycle. You pull those individual organic compounds out, you stick amino groups onto them, you can make amino acids. So here are the 20 different amino acids that are typically used to make protein. They're just modifications of organic compounds. And then if we stick those amino acids together, what do we build? Polypeptides. And if you put them together, so you can make polypeptides at least, and possibly proteins. So some proteins we know have more than 1 polypeptide. Some proteins are functional just as a polypeptide chain. We can make proteins. Consequently, although it seems like we just look at a few metabolic reactions, the ones we look at are really central to metabolism, both catabolism and anabolism. We looked at them initially, we were talking about organisms getting energy. Then you can go back and we talked about chemoheterotrophs using organic compounds for both an energy source and a carbon source. This biosynthesis stuff is what we're talking about. They're using these organic compounds to make intermediates that then they can use to make other kinds of things. So this tells us if organisms can make these other kinds of organic compounds, then it stands to reason they can also take these other organic compounds apart and get energy from them. And in fact, they can. So this is talking about triglycerides and phospholipids being taken apart. You take the fatty acids off the glycerol can again be converted to DHAP and will be catabolized following the rest of the glycolysis pathway. The fatty acids, these long chains of carbons, they're actually hydrocarbon chains, those can be cut into 2 carbon units using a process called beta oxidation. So beta oxidation cuts these long chains of fatty acids into 2 carbon units and puts oxygen on them so that they are again acetyl groups. Those can bind with coenzyme A and you get acetyl-CoA. So lipids can also be used as an energy source. Of course, the amino acids from proteins can also be used as an energy source. All you have to do is pull the amino groups off, and you have organic acids that can either be catabolized using glycolysis, or they can be catabolized using the Krebs cycle. Typically, when we think about organisms using organic compounds as a carbon source and energy source, we're talking about carbohydrates, lipids, and proteins. Nucleic acids can be taken apart, but nucleic acids are there's a very limited supply of them in cells. There's not much there. Nevertheless, they're extremely important. So I just stuck this picture on here. Because this is an aerial view of something that's actually happening, what do you suppose is going on there? Actually, I saw a commercial about this just recently. So this is probably a petroleum company that is endeavoring to contemplate something other than fossil fuel as something that could be used to make petroleum type products. So they're growing algae. They're either algae or cyanobacteria. I'm not real sure because of these pink ones here. So it's like, what are these? I don't know. Some other kind of thing. But anyway, very large areas being dedicated to growing organic compounds. I don't know how efficient it is, but it's kind of interesting. So this is being done. Let's face it, fossil fuels were originally organic compounds produced by living organisms. They were just produced a long, long time ago. And now we're just burning them up and releasing the CO2. Okay. Stop, that's the end. So now we're going to shift gears almost entirely because here we were talking about metabolism. Metabolism is the chemical reactions are catalyzed. The 2 types of catalysts we talked about are enzymes that are proteins and therefore made of amino acids. And we talk about ribozymes, which are RNA molecules and therefore made of nucleotides. In either case, which enzymes a cell can make or which ribozymes a cell can make is totally dependent upon the genetic composition of that cell. So now what we're going to do is shift away from metabolism and start talking genetics. First I have to find it. Might be a good trick because I don't have my glasses on. Oh no! Okay, so part of this presentation is going to be review, because we've already talked about nucleic acids when I was introducing organic compounds in general. So review is always good because it's part of the learning process. So this is DNA, RNA, replication, and transcription. What is DNA? Yeah, deoxyribonucleic acid. Don't ever forget what those letters stand for. So you won't forget DNA. The Na is nucleic acid. Nucleic acids are made of nucleotides. I couldn't believe how many people mixed up nucleic acids, nucleotides, amino acids, proteins on exam 1. If you need to go back and review that very basic chemistry, you need to know. And it needs to be just immediate when you see those terms, which ones are we talking about. Don't mix them up. So DNA. Na is nucleic acid. So it's deoxyribonucleic acid. I'm making the letters violet in here because when we were talking about the groups of organic compounds, I said nucleic acids are going to be purple or violet or whatever you want to call it. So it's a type of organic compound. It's a polymer, long chain molecule. It's made up of smaller units, and the smaller units again are nucleotides. Nucleotides. Each nucleotide contains a sugar, If we're talking about DNA, the sugar is deoxyribose. It also contains a phosphate group and one nitrogenous base. Each nucleotide has one nitrogenous base, but the nitrogenous bases aren't always the same. So in this case, we have a sugar, the nitrogenous base is adenine, and we have a phosphate group. That is a nucleotide. If you wanted to name it, it's adenosine monophosphate. That's what you would have before you got to ADP, before you got to ATP. A single nucleotide has just one phosphate. So, this is really a review. Nucleotides are the building blocks of nucleic acids, but when I was talking about organic compounds, remember I said they had other functions within cells? It's like, what were nucleotides used for? Besides nucleic acids. They're used to make coenzymes. So NAD, FAD, now you have a better idea of what coenzymes are. They're enzyme helpers. Again, on this last quiz, so many people wanted to tell me that a coenzyme is an enzyme. No, it isn't. A coenzyme is a non-protein organic compound. It can't be an enzyme. Enzymes are proteins. It's a non-protein. But a coenzyme, NAD, is nicotinamide adenine dinucleotide. FAD is flavin adenine dinucleotide. So they are made from nucleotides. They're also derived from vitamins, but anyway. So that was one function. The other function is ATP. Stick 2 extra phosphates onto adenosine monophosphate and you have adenosine triphosphate, which is a high energy compound, the most important energy currency within cells. The last function we haven't talked about yet, we'll get to. Nucleotides can be regulatory molecules. So we're going to talk about cyclic AMP later. We'll get to that later. There's also cyclic GMP. Okay, so coenzymes. The cyclic AMP is a regulatory molecule, and then the high energy compounds. ATP is one, but there's also GTP, CTP, UTP, etc. Okay. So DNA, this can be abbreviated, or I should say the nucleotides that are used to build DNA are abbreviated DNTP. The small letter D stands for deoxyribose, so that you know what the sugar is. So a DNTP is a nucleotide used to build DNA. If you were making RNA, that NTP would be preceded by the small letter R, RNTP. So this is a DNTP because the sugar is deoxyribose. And how do we know? If you look at the bottom, and instead of having 2 hydroxyl groups down here, there's only 1. If this was ribose, there'd be 2 hydroxyl groups. That's going to be significant to some processes we'll talk about a little bit later on. So there's a sugar missing from deoxyribose. When I was introducing carbohydrates, I said the word carbohydrate means hydrate of carbon. And supposedly for every carbon atom in a sugar, there's a water molecule. Not that one, not for ribose. Excuse me, not for deoxyribose. It's missing 1 oxygen. Well, anyway. So that's the important part of the sugar. So the high energy compounds, the DNTPs, provide the energy that's necessary to build the covalent bonds between nucleotides when nucleic acids are being made. And those covalent bonds are called what? So between nucleotides, when you're building nucleic acids, you're connecting nucleotides together. What are the covalent bonds that hold them together? Phosphodiester. Hydrogen bonds are covalent. Hydrogen bonds are weak bonds. Covalent bonds are strong bonds where the atoms are actually sharing electrons. So they're called phosphodiester bonds. That's important. And again, this is kind of review. Oh, and yeah, the process used to make them, dehydration synthesis. So remember, you have to take a water molecule out every time. This is all review. So the phosphodiester bonds occur between the sugar of one nucleotide and the phosphate group of the next, and they're forming what we call the backbone of the nucleic acid. Sugar phosphate, sugar phosphate, sugar phosphate. And then the nitrogenous bases are kind of sticking off of there like fins. So here you see where the bonds occur. The sugars are the little box-like structures. these little box-like structures. These are the nitrogenous bases. If there's two rings, what kind of a nitrogenous base is it? 2 rings is a purine. If there's only 1 ring, the nitrogenous base is a pyrimidine. So we have purines and pyrimidines. So the 4 possible bases of DNA. So I said they're purines and pyrimidine. Adenine is one of them. What are the other 3? Guanine, cytosine, thymine. You're building DNA. Adenine, thymine, cytosine, guanine. Or, the reason they're in the order that they're shown here is because that little diagram had them in that order. It's like, yeah, okay, fine. Thymine, cytosine, adenine, guanine. Which ones are purines? 2 rings. Adenine and guanine. Adenine and guanine. Those are the purines. Here's a way to remember this if you're familiar with UC Davis. UC Davis, what's the mascot? Aggies. Aggies are pure. You remember Aggies are pure. You got it. Adenine and guanine. If you think otherwise, well anyway, it doesn't matter. Cytosine and thymine are pyrimidine. The other way to help remember is cytosine and thymine and pyrimidine all have Y's in them. So all the Y words go together. And that works just fine until you get to uracil, and uracil doesn't have a Y, but it's still a pyrimidine. Okay. Now, the other thing we learned about those nucleotide chains is that in the DNA molecule you have 2 nucleotide chains, and they're oriented upside down relative to one another. So we said they're oriented upside down relative to their 5' and 3' ends. So the end of the chain that has the phosphate group is the 5' end. The end that has the hydroxyl group is the 3' end. So this is called the 5' because the number 5 carbon has the phosphate on it. This is called the 3'. The number 3 carbon has the hydroxyl group on it. So there's an orientation to each nucleotide, and then there's also an orientation to a chain of nucleotides. Phosphate at the 5' end and hydroxyl group at the 3' end. So here we see a nucleotide strand. Phosphate at the top, that's the 5' end. Sugar at the bottom, well, yeah, it's a sugar, but it's the hydroxyl group on the sugar that is the 3' end. And then the different bases are sticking out to the side. You can see lots of different ways to diagram DNA. So again, cellular DNA molecules, typically 2 strands. And the reason I'm making a point that it's cellular DNA molecules that are typically 2 strands is because some viruses can be single-stranded DNA. So cellular DNA is typically 2 strands. Viral DNA can be 2 or 1, doesn't matter. And apparently there's some evidence that some cellular strands of DNA have more than 2. So typically we're talking about the most common form of DNA, 2 strands. These 2 strands are held together by relatively weak bonds. What kind of bonds? Hydrogen bonds. And hydrogen bonds are occurring between the bases. This diagrammatic representation shows you how many hydrogen bonds. So that doesn't mean A equals T. It means between adenine and thymine there are 2 hydrogen bonds. Between cytosine and guanine there are 3. So it's different numbers of bonds. And here you can see thymine, adenine. The hydrogen bonds are here. So there's 2 bonds between those. And then cytosine, guanine, 3 hydrogen bonds between those. This influences the stability of DNA. So if you have a DNA molecule that has more cytosine guanine pairing, they tend to be more stable. It's more difficult to break them apart because you have 3 hydrogen bonds between them. So we have our hydrogen bonds. Again, notice the number. The hydrogen bonds are always between a pyrimidine from one strand and a purine of the other. But it's more than just pyrimidine to purine. One of the reasons for that pyrimidine-purine bonding is because purines have 2 rings, pyrimidines have 1. So between the 2 strands, there's always 3 rings. 3 rings per rung, if you will. but it's more specific. So it's not just any pyrimidine. It's always adenine to thymine, cytosine to guanine. So there's 3 rings per rung. Consequently, if you have lots and lots of hydrogen bonding, which you do in a DNA molecule, even though the hydrogen bonds individually are weak, you have so many of them that it holds the DNA together fairly well. DNA is a pretty stable molecule. On the other hand, it's pretty easy to break them too. In the laboratory, we just heat them. Get them hot, they come apart.
Literally, everyone's not. It's over. Yeah.
So if you go out, you can get back in. Sorry. So this again, this is all review, right? There's all those hydrogen bonds. So when we say the 2 strands are complementary together, this means that the 2 strands make the whole. The 2 halves make the whole. But if you know the nucleotide sequence, the base sequence of one strand, you automatically know the nucleotide sequence of the other. This becomes really significant later on in here because we're going to be looking at nucleotide sequences as a method for identifying bacteria. And we have to remember that the computer knows that if you have one set of letters for one strand, you automatically know what the flipped version of that strand is, or the complement. It'll be called a reverse complement. Anyway, the 2 strands are complementary. Adenine always pairs with thymine, cytosine always pairs with guanine. So if you have that sequence, you know the complement is complementary bases. So you automatically know. And again, this is very significant. The other thing is these 2 strands are anti-parallel. So where one is 5' at the top, 3' at the bottom, the other one is the other side up. 5' at the bottom, 3' at the top. So anti-parallel means they're side by side, but upside down relative to one another. It's hard to do. This is also going to be very significant when we start looking at the process of replication. How does DNA reproduce itself? And it's important that one strand is read in a continuous sequence, and the other strand is going to be built in a whole bunch of fragments because it's upside down or anti-parallel. The other thing about that duplex is it's not straight. If you think of the DNA being like a ladder, it's twisted. So it's more like a spiral staircase. There is a specific pattern to that twisting. The model that's out in the hallway isn't right because it doesn't have the right pattern. And I can show you a kind of a representation of it. Using this very high-tech model. Hold on to the bottom. Now, from some distance, what you'll notice is that this is 2 slinkies stuck together. But what I'm trying to show here is that there is a variation in the twist. So what you see is a rather large space, and then a tiny little space. Then a large space, and a tiny little space. Large space, little space, large space, little space. That's how the helix, the 2 strands of the DNA molecule are arranged. So in that helix, there are big spaces, little spaces. Do you get to draw that? No. I'm not trying to draw that on the board. I can't ever do it. But all of these look like it's uniform, and it's not. It's like that. So those are called major grooves and minor grooves. There's so many major grooves and so many minor grooves. And so what's more important is that the base sequence, the nucleotide sequence, adenine, thymine, cytosine, guanine, those are going to determine the anatomy and the physiology of an organism. because the DNA determines all the RNA molecules that a cell can make, some of which are ribozymes, and the DNA determines all the proteins that a cell can make, all the polypeptides. So it's an extremely important molecule, even though it makes up a very small percentage of the organic content of a cell. Nucleic acids are minimal relative to organic content. We're going to see the term gene, and I can give you one definition for gene now. This is a kind of an interesting term because the term was coined prior to people really having any idea what DNA was. A gene was considered a segment of heredity, or a unit of heredity, and it took a long time for people to figure out what it was really, because people used to think a unit of heredity was protein. It couldn't possibly be nucleic acid. They were too simple and there wasn't enough of it. Turned out to be nucleic acid indeed. So we can define genes as segments of DNA. If we're talking cells, that's true. If you're talking some types of viruses, genes can be RNA, because viruses can have either type of nucleic acid, either DNA or RNA. So genes are segments of DNA. DNA, we know, coiled up around histone proteins in eukaryotic cells, and then the histone proteins combining to make nucleosomes, and then the nucleosomes piling up to make chromatin, and then there's heterogeneous proteins, enzymes associated. So the DNA does form chromosomes, but chromosomes are very tightly wound chromatin. There's more to it than just DNA. So we say DNA is the genetic material of all types of cells because genes are DNA. And now we're going to talk about RNA. Again, part of this is review because we've seen it before. So RNA, that stands for
ribonucleic acid.
Ribonucleic acid. Again, Na is nucleic acid. The difference between DNA and RNA, in part, is the sugar. So RNA, ribonucleic acid, again, it's a long chain molecule made up of nucleotides, but not nearly as long as DNA. So DNA molecules are huge relative to RNA. The sugar in RNA molecules is ribose. You've got 2 hydroxyl groups instead of only 1 at the bottom of that sugar. So on the number 2 carbon and the number 3 carbon, we have hydroxyl groups. That's how we distinguish ribose from deoxyribose. And one of the bases is different. So DNA has adenine, thymine, cytosine, guanine. RNA has adenine, uracil, cytosine, guanine. And actually, thymine is 5-methyluracil. So thymine and uracil are the same, except thymine has a methyl group and uracil doesn't. So when we're building the covalent bonds to build RNA, it's the same as when we're building the covalent bonds to build DNA. The high energy compounds that provide the energy to make the covalent bonds are NTPs, nucleoside triphosphates, DNTPs for making DNA, RNTPs for making RNA. And the bonds again are phosphodiester. And to make them, you have to remove water, dehydration, synthesis. So that's all review. Now RNA molecules also have an orientation because they're going to have phosphate on the number 5 carbon and a hydroxyl group on the number 3. It's just that RNA molecules are typically just single strands. Now, if you were talking about RNA in a virus, some viruses have double-stranded RNA. And if they do, the 2 strands are complementary and anti-parallel. So RNA can behave just like DNA. So this is showing RNA with the 5' end with the phosphate, the 3' end with the hydroxyl group. And again, RNA molecules are much smaller than DNA, much, much smaller. And whereas DNA is the genetic material within cells, RNA molecules have a wide variety of functions. Some of them are ribozymes, some of them are not. But you're going to get to see a variety of functions of RNA molecules. Later it says, So there you see a diagrammatic representation of DNA. Again, this is cellular DNA. It's a double helix. And they didn't even show the major group, minor group. They exist, really they do. And then the RNA molecule, which is a single strand. Okay, so that's kind of review with a few extra details thrown in, but we covered most of them before when introducing the organic compounds. So now we're going to talk about DNA replication. We know a little bit about DNA replication because I mentioned some things when talking about how bacteria undergo binary fission. So DNA replication is the process used to make more DNA when DNA molecules reproduce. This process is extremely complex, and what's kind of interesting to have been involved with biological sciences for quite a long time, it's interesting to see how knowledge has shifted over time in terms of what goes on here. Used to be there was just one enzyme that was named in association with replication, and the enzyme was called DNA polymerase. A DNA polymerase builds a polymer of DNA. And if you were building RNA, you used RNA polymerase. It's like those were the only enzymes. Now we have a whole bunch of enzymes. It's like, yeah, of course. There's more known, and so what it tells you is you're going to get a somewhat simplified version. Yes, because at some point, I just stopped and said, okay, that's enough. This is an introductory class. The information that has been building on DNA and RNA and what's going on with these molecules is quite amazing, but a little overwhelming for this class. So we're going to look at replication in a somewhat simplified way. One thing about DNA replication is it is semi-conservative. What that means is you have two strands of DNA. When replication occurs, the two strands separate, and each old strand serves as a pattern or a template for building a new strand. It would be like if I had a zipper and I could unzip it, and then somehow the teeth on this zipper encoded complementary teeth for a new zipper. And they came in as individual units, and then they all got stuck together. What that means is every time DNA replicates itself, the two strands come apart, and you have the new DNA that's built is half old and half new. So then if you think about how old DNA is, you don't want to think about it too hard because it's really, really old. At least parts of it are. It's always semi-conservative. You keep one half. So how old is it? Yeah, old. Really old. The other thing about replication is we're going to be looking at one of the most common forms of replication. I probably should have said that when talking about glycolysis too. We're looking at one of the most common forms of glycolysis. It's not the only one. We're looking at a particular type of replication. It doesn't always go the same way, but we're going to talk about the one that's most commonly used. So when we think about replication, what's required? Number one, you have to have an existing DNA molecule because, as I said, replication involves two strands of the DNA molecule coming apart. So you have to start with DNA so that the two strands can come apart. Got to have DNA as a template. The next thing you have to have is enzymes. Used to be just one, DNA polymerase. Now we're going to find out, oh no, there's a whole bunch of enzymes. So when talking about chromatin in the eukaryotic cell, chromatin is a combination of DNA and proteins. Some of the proteins were histones, some of the proteins were heterogeneous, so a lot of different kinds. Well, some of those are enzymes that are involved in DNA replication. Some of them are enzymes involved in making RNA, but we'll get to that later. The third thing that's required is energy, because making DNA replication, is synthesis. It's a building process, and building those phosphodiester bonds, you have to have energy. Where did the energy come from? ATP, yes, but DNA isn't all adenine. So the energy comes from DNTPs, ATP, GTP, CTP, TTP. Every one of those things can be a high energy compound. So they are DNTPs. That's where the energy comes from. It's a very energy expensive process making DNA. Every single phosphodiester bond requires one high energy compound. Now, within living cells, typically DNA, again, we're talking about the most common form of DNA replication, it starts at a point that's called the origin. And if you were looking at a prokaryotic cell where you have a closed, covalently closed loop of DNA, there's typically only one. So there's one spot where DNA replication occurs. That's called the origin of replication. And from that point, replication is going to proceed in both directions around the loop. Here you see a replication in process, so that here's the double helix, and here's double helix on the other side. If the origin of replication was here, so we started replicating, we're going to be replicating in both directions away from that point. This diagram has quite a lot of information on it because it has enzymes and it has other things that are going on there, which is the leading strand, which is the lagging strand. We'll get to that in a bit. So for prokaryotic cells, again, they have covalently closed circular DNA molecule. Replication starts at the origin and goes around both ways. The origin is usually called ORI-C. So if you see ORI-C someplace written, you can say, oh, that's the origin of replication. Replication starts there and it goes around. If we're talking about a eukaryotic organism, eukaryotic organisms have linear chromosomes. And if you've ever wondered, does a chromosome start replicating at one end and replicate the whole length? The answer is no. Eukaryotic chromosomes also have origins of replication, but they have more than one. So they'll have a whole bunch of them. If we're looking at this little segment, this is a eukaryotic chromosome. Well, probably not accurate because there's more origins. But anyway, at each origin, each ORI, replication will start, and again, it goes in both directions. So here it's just begun, and here it's gone further. Ultimately, you'll have two strands. So it's rather complex. So again, the origin of replication for bacteria such as Escherichia coli, it's called ORI-C. When you see it in these diagrams, it looks like a single point, but actually the origin of replication is quite complex. This is the part when I say you're getting a very simplified version. It's because we are not going to look at all the details of that origin. It's 245 base pairs in length. Wow. When you measure DNA, DNA is measured in base pairs. So adenine, thymine, cytosine, guanine, the rungs of the ladder. So something that's 245 base pairs long, that's a pretty good section of DNA. It's not just one spot. And that's because there's a bunch of things that have to go on there. It is significant though. Remember when I was talking about binary fission, when bacteria undergo binary fission, and they can undergo fission every 20 minutes, even though it takes E. coli 60 minutes to replicate the entire chromosome? It's because they run multiple cycles. So they'll start replication at the origin, it begins to proceed around the loop, and then they'll start another cycle, which begins to proceed, and then they'll start a third cycle. So they're running 3 cycles at once. Now, the DNA is going to interact with a whole variety of proteins. Some of them are enzymes, some of them are not. You'll see in this diagram, there are single-stranded DNA binding proteins. Those apparently bind to the DNA. Once the strands come apart, they're going to bind to it to prevent it from tangling up or to stabilize it. So, again, this is kind of a simplified version, but you need to know what's involved. So step 1 of replication. The DNA double helix has to separate. As I said, in the laboratory, we can make DNA strands come apart just by heating it. But getting DNA hot in a living cell is not the best thing to do for the cell. So that's not going to happen in the cell. Instead, the DNA separation of the 2 strands is going to involve enzymes. So it comes apart in just a little small section. The enzymes involved are called helicases and gyrases. So helicases apparently break the hydrogen bonds and allow the strands to come apart. But remember, the DNA is twisted. It's a helix. And consequently, have you ever taken a rope that is twisted strands and start pulling the strands apart? And you watch the rope and it just starts spinning like crazy. So the helicases break the hydrogen bonds to let the strands come apart, and these other things that are called gyrases or topoisomerases play a role of preventing this twisting. They apparently break the DNA and reattach, break, reattach, break, reattach to keep it from getting all tangled up. So the helicases are motor proteins. They kind of move along the DNA. They break the hydrogen bonds, let the strands come apart. And then those single-stranded DNA binding proteins jump on there to protect the single strands. And the gyrases prevent the DNA from twisting too much or messing up the replication process. And they also move along ahead of what's called the replication fork. So as this is a view of replication. In this case, the replication is going this way. So the process is going along in this direction. This is our topoisomerase that's kind of preventing the DNA from getting tangled up. And the helicase is coming along here, and then these are the DNA binding proteins. Where you see these two strands separate, this is called the fork. So there's a replication fork. From the origin, there's going to be two replication forks moving away from one another in opposite directions. But a fork is just this. There's the replication fork. Yeah, okay. So the next thing that's going to happen, if we're going to do DNA replication, is there are these little short nucleotide chains called primers that are going to come upon. They're going to attach to the DNA. Part of the reason for the primers is because the primary enzyme associated with DNA replication is DNA polymerase, but DNA polymerase cannot bind to a single strand of DNA. It can't do anything. And so we have to hook these little short sections of DNA called primers onto the DNA to make it a duplex. What's kind of interesting about this is that the primers are made of RNA rather than DNA. This has to do with, again, the enzymes that are capable of it being involved in this process. The enzyme that builds RNA polymerase, can bind to a single strand of DNA, but DNA polymerase can't. So the RNA polymerase is going to build these little primers. When we're talking replication, although technically the molecule is a DNA-dependent RNA polymerase, we're going to call it primase. If it's building a primer, we're going to call it primase so that we don't get it confused with RNA polymerase later. These primers are relatively short, around 18 to 20 nucleotides long. And again, the enzyme, even though it is a DNA-dependent RNA polymerase, we're just going to call it primase. So primase is the enzyme that builds those guys. And these are very complicated enzymes. So they're not simple enzymes. They have multiple segments to them. And this, again, is kind of interesting because if you look at older references, they may say, this enzyme has this many pieces, and then you look at a newer reference and it's like, no, it has more than that. This is something that's constantly changing as more information is gained. But that gives you an idea. This thing is very complicated. It's an enzyme complex. So once you have the primers in place, the next step is to add new nucleotides to the primers. They can only be added at the 3' end. And again, this is because of the enzyme. DNA polymerase is only capable of adding nucleotides to the 3' ends. It's going to be catalyzing the formation of phosphodiester bonds, but it can only do so in one direction. It's always connecting nucleotides to 3' ends. So it's building from 5 to 3. So here we see DNA polymerase. This strand that is being built is the red one. So here's the 5' end down here. This is where it started. And it's building by adding nucleotides along, but it's building from 5 to 3. So the 3' end here, this is where new nucleotides can be connected. This is a real simplified version of it because the DNA polymerase is shown being fairly simple. if the origin was somewhere in here, this strand is being built in this direction. Meanwhile, this strand is being built in this direction. Here's another DNA polymerase. Here's the 5' end of the red strand. Here's the 3' end, and it's the 3' end where the nucleotides are being stuck on there. Turns out this is rather significant, because if you follow any single replication fork, one strand is going to be built as a continuous sequence because it's just constantly being built from 5 to 3. But the other strand is the other side up. And if you started building this strand here, as you go this direction, this strand is going to be just a single strand of DNA for quite some time until you can put a new primer on it. Turns out that the enzyme that can build the primers can only attach at certain locations. Well, we'll get to that in a second. So the primary enzyme involved in making DNA, again, is DNA polymerase. Now, it is specifically a DNA-dependent DNA polymerase, and the significance of this is that if you were looking at viruses and you were looking at them building DNA, sometimes they can use an RNA-dependent DNA polymerase, which is also called reverse transcriptase. So in cells, it's DNA-dependent. That just means it has to have DNA as the template. If it's going to build DNA, it has to have a DNA template. Most of the time, we just call it DNA polymerase. And again, it has limitations. One, it can only bind to a duplex. That's why the primer was necessary in the first place. You can't get DNA polymerase to attach to a single strand of DNA. It can't do it. You have to have a primer. So it can only bind to a double-stranded DNA, and it can only build in one direction, from 5 to 3. Actually, as far as I know, that's true of all the polymerase enzymes. They can only build in one direction. So they're always sticky nucleotides on the free 3' end. That's where they build their phosphodiester bonds. In prokaryotic cells, there's more than one DNA polymerase. So as I said, it used to be DNA replication, there was only one enzyme and it was DNA polymerase. And oh, by the way, there was only one of them. Turns out that's not true. There's actually 3 different versions. So there's DNA polymerase 1, 2, and 3. The primary builder is DNA polymerase 3. So on quizzes and exams, that's the one I'm going to be asking about, the primary builder, DNA polymerase. You can just call it DNA polymerase. But it is DNA polymerase 3. Those other DNA polymerase molecules have slightly different functions. So they are going to be involved in the replication process, but they're not the primary builders. This DNA polymerase 3 can add nucleotides at a rate of about 1,000 per second. That's kind of staggering when you think about it, and think about the utilization of ATP molecules, because all the DNTPs are equivalent energy, high energy compounds, ATP, GTP, CTP. If they're building phosphodiester bonds at a rate of 1,000 per second, that's pretty fast. A lot of energy being utilized. Consequently, they have to have an energy source to replace. I have a bad feeling there might be a film coming up, and I don't have my sound things set up here. Well, we'll see what happens. Okay. So the enzyme builds very rapidly, but it can only build in one direction. So it can only add nucleotides to a free 3' end. That is the end of the primer. If the DNA strand is my arm here, the primer's here, this is the 3' end of the primer. So it can only build on this, it's going to go that direction along the template. So you see lots of different diagrammatic representations of this whole thing. So here you see, again, a replication fork, and it's showing the helicase taking the strands apart, the DNA polymerase come along and building. But if you have 2 strands, and one is built as a continuous sequence, 5 to 3, the other strand's the other side up. And consequently, you can't build a continuous sequence on that side. This is showing what's called a replisome. So the replisome is all these parts, the helicase and the topoisomerase and the DNA polymerase and so forth, and never mind. Like I said, there's a lot more detail to this stuff than you need to get into. This is an introductory class, so we're not going to go that far. So if you start at an origin, as I said, you're going to have 2 replication forks moving in opposite directions. Typically, one strand of the DNA molecule is referred to as the leading strand. and the other is referred to as the lagging strand. And the reason for this is one is built as a continuous sequence, being built from 5 to 3. But because the other strand is upside down, you have to start building. It's going to unzip for a while, and then you start building, then it unzips some more, and then you start building. It's built in a series of segments. So the leading strand is the one that's built as a continuous sequence, and the lagging strand is built as segments. However, if you have a DNA molecule, you're going replication fork this way, replication fork this way. The strand that is the leading strand on this side is the lagging strand on this side. So there isn't really one leading strand, one lagging strand. Those are terms that only refer to what's going on at any given replication fork. And again, lots of diagrammatic representations of what's going on. The DNA polymerase enzymes, once they're attached, they're going to be moving away from the origin in opposite directions. But because of this addition of nucleotides only happening at the free 3' ends, the DNA is built from 5 to 3. Templates are upside down. Consequently, one gets built as a continuous sequence, that's the leading strand, and the other, the lagging strand, is built in a series of sections. And these sections are called Okazaki fragments. They're named after Dr. Okazaki, who apparently discovered them. So Okazaki fragments are these little fragments of DNA that are built on the lagging strand because the strands are upside down or anti-parallel. This is showing building of the leading strand, where this is the primer, the leading strand primer attached. This was free 3' end. You could add nucleotides. It's building continuously this way. Now, if you can imagine, the unwinding was happening, also going in this direction, but we had to put a primer on and build here, and then unwind, put a primer on and build this way, unwind, put a primer on and build this way. These are the Okazaki fragments. So each Okazaki fragment begins with an RNA primer. This diagram makes it look like they're really short. They're not really short, but you can't fit them into a diagram if you make them big. I have to stop. Okay, so you guys have a take-home quiz next time. The take-home quiz covers photophosphorylation, photosynthesis, biosynthesis, and the beginning of DNA replication. We didn't get very far, but we got the Okazaki fragments. I know you guys are thinking, oh great, we don't have to worry too much about this quiz because it's take-home. And that's true, because really what I want you to do over the weekend is study for the lab exam, because that lab exam is 150 points. That's a big deal. But don't forget to study at some point photosynthesis, biosynthesis, and this DNA and DNA replication. Okay. So we have a break and then we have a lab quiz.
No, don't be. I just go back because I know it's distracting that I'm like feeling so wiggly.
It's more distraction for you. So I have my own space.
Yeah. Afterwards.
Yeah.
However,
The phone guys.
so we may have two spaces.
Yeah.
Not yet, not yet.
Okay.
Yeah, I don't wanna throw somebody out like this, I'm not pretending to read this stuff.
Yeah.
I was like the first 4 or 5
Oh, it's so annoying.